Okay, people are still joining. The recording has started. Good evening, everybody. Welcome to another session of Aging and Regeneration. Today, we'll be talking about Parkinson's disease, and we had planned for mitochondrial dynamics, but I think it won't be possible today. We'll take up this part on Saturday. So it will be probably a long, lecture that day. So, anybody who knows about Parkinson's, what happens?
a disease a disease which
It's
causes in a disease which causes inhibition of dopamine levels. That
That is correct. So
So
essentially,
Parkinson's basically it's a palsy or it's a palsy type of disorder. It has mental effects also. I've seen with my own eyes, and uh, it it actually makes the person look a bit lethargic. The person gets immunologically weak as well as lethargic. He or she can also be a subject to fatigue also, and
correct.
these are the symptoms that
Correct, correct. A lot of things happen actually, but since you said it is a kind of disease which looks, which is similar to palsy, it was first reported as shaking palsy by a person called James Parkinson's. That's why the name comes in as Parkinson's disease. So it was first reported in around 200 years ago in 1819, and this is how it looks like. So the first essay came out in 1817, and then the disease was named. And essentially, there are involuntary tremulous movements, which we call also as palsy. So uh, it was later in 1910 when the dopamine secretion was linked with Parkinson's disease. And as you can see somewhere here, and here, and they found out about this disease. So if you want to find out more, there is a website link here, you can find it out from there. So it's a slow progressive neurodegenerative disease with a high incidence in aged people. So aging is one of the greatest risk factor in Parkinson's disease, and if a person is aged above 60 years, there is a high chance that the person will get Parkinson's. So it is second most common neurodegenerative disease just after Alzheimer's, which is also very similar, and it can be classified as familial or sporadic. So familial is when you have it from your family, or sporadic if you have it, just from the environment. So familial Parkinson's, it contributes to about only 20% of the Parkinson's cases, whereas sporadic is around most common. It's the most common way you get Parkinson's. So how do sporadic Parkinson's happen? We don't really know. And that's an active field of research still going on. So we'll see what happens. So some of the symptoms, cardinal symptoms are like tremors, which was characterized in the year 1820. Sorry, in the 19th century, early 19th century. Um, so that what happened. Bradykinesia, it's slowness of movement, and akinesia, inability to initiate movement. These kind of things are observed. There is postural instability and also rigidity, which is observed. So if we talk about Parkinson's disease, it happens essentially due to a part in the brain which is called the substantia nigra. Can everybody see this?
yes, sir.
Okay, so generally
yes
what
sir.
happens in both, in the case of no, in a normal person, the substantia nigra, as you can see, it is a little bit dark in color. This is because of the accumulation of the pigment called melanin. Here is where dopamine is also involved. Dopamine secretion is also there. It's responsible for dopamine production. Now, when there is, accumulation of the alpha-synuclein protein, which is a disordered protein, which we see in substantia nigra, as you can see clearly, the melanin is no longer there, and the substantia nigra is deformed in the left hand side. You can see that compared to a normal, as, substantia nigra. So this is what happens, and there are motor deficits because of this. So today's definition is of Parkinson's disease is a chronic. It's a chronic progressive neurological disease which happens in later life, and it is linked to decreased dopamine production in the substantia nigra and is marked especially by tremor of resting muscles, rigidity, slowness of movement, impaired balance, and shuffling gait. All these things are observed. Some non-motor symptoms are also there, like there is olfactory dysfunction, constipation, drowsiness, as one of you already mentioned. There is pain, anxiety, sleep disturbance, and depression also because of this. So when we move forward, due to the familial form of Parkinson's, that happens due to this would be familial, not familiar. Familial forms of Parkinson's, it happens due to the mutations in these genes. So like SNCA is for production of alpha synuclein, LRRT2, which is a leucine reach repeat kinase, which is involved in autophagy lysosome pathway. Then there is ubiquitin proteasome system, which is affected. We have already studied about ubiquitin proteasome system in the last lecture. And then Parkin, which is an important ubiquitin ligase in mitochondria. We'll be studying about the function of Parkin and PINK1, both of these in detail. And also DJ1, again, it's involved in mitochondrial function. So what usually happens in the disease is there is enhanced production of alpha synuclein, and the degradation is not normal. So all hallmarks of aging, they are usually seen in Parkinson's disease. That's why it's a great example of um, of aging, basically. So you can see mitochondrial dynamics is affected, the ubiquitin proteasome system, that means the proteostasis is affected, vesicle trafficking is affected, and all these kind of things, and all the ones which are given in the boxes, all these proteins are essentially chaperones. We already talked about it. So they are involved in protein folding. So those proteins, they have an effect. So we will see one of the hallmarks of this disease is essentially um, well it's these intracytoplasmic inclusions. So there is formation of Lewy bodies. Fine. These Lewy bodies mostly contain alpha synuclein aggregate. That is what they have. And um, they do not only have the full length alpha synuclein, but also sometimes fragments of alpha synuclein. So uh, probably this fragmented alpha synuclein, it causes the toxicity, but it is not known yet. Okay, so this is an active line of research, and what causes this fragmentation of this protein, that is also not known. So uh, then comes about the function of alpha synuclein. So again, if we talk about the function, then you can see again the function is not clearly known about this particular uh, protein, but there are some evidences that alpha synuclein has some role in dopamine secretion. So as you can see, this is the SNCA gene in the chromosome 4 that synthesizes the alpha synuclein. If it is folded properly, earlier it was thought that it exists as a monomer, but now recent studies they show that alpha synuclein, it exists as a tetramer. So if there is any misfolding, there are monomers which are present, which form oligomers, which form protofibrils, and then these protofibrils mature to form Lewy bodies. This is the process, essentially how it happens. And uh, which form is most toxic, whether it's the oligomers or whether it's the protofibrils, that is not known. So if there is enhanced expression of SNCA, if it has been experimentally done, so we see that it creates a problem. So if these chaperones like the HSP70 proteins or all these heat shock proteins, these are all chaperones mostly. So if they are overexpressed, then we see that the problem is somehow solved. So clearly, there is a problem in folding of this protein that causes the problem. Um, Later, few studies also found that alpha-synuclein has some role in vesicular trafficking of dopamine, but again, we don't know what happens. So this is how it looks like. So the same figure from a different
Can you
paper.
explain? Can you explain the sir? Sir,
Sorry. Yes.
hello. Sir, can you please explain that that chaperone part once more? Like if the chaperone expression
If
increases, then what
if
happens?
if chaperone expression is increased, it has been experimentally seen that that reduces the symptoms of the disease. So that has been observed. So that is again an experimental setup which we, so essentially misfolding is the problem. If they do not fold properly, if alpha synuclein doesn't fold properly, it will form oligomers and protofibrils and then Levy bodies. So it's some
Okay, sir.
way related to
So,
proteostasis. So we have seen that proteostasis is again an
yes,
hallmark
sir.
of aging. So yeah. So this is how it is linked. Again, the degradation, it happens through all the three routes, like macro autophagy could be there, cell mediated autophagy could be there. Just a minute, just a minute, just a minute. I have a call. Yeah, I'm really sorry for that. Anyway, where were we? Okay, so the degradation happens through all the three routes: macro autophagy, cell-mediated autophagy, and ubiquitin proteasome system. We'll be talking about all of these in detail. So this is another figure which shows the role of mitochondria with, this alpha-synuclein. Again, something different. So here also we will talk about this in detail actually. So when we talk about the degradation of alpha synuclein, so you know uh, the degradation could happen due to a lot of things. So it could be ubiquitin proteasome system, it could be cell mediated autophagy, it could be degraded in the cytosol using enzymes like caspases, and there could be macro and micro autophagy which could also work for degradation. But all these things are usually affected when alpha synuclein is uh, accumulated. So we'll see how that happens. So when we will see each part together. So in a normal condition, when alpha synuclein is there, the chaperones and co-chaperones, they fold it properly, and then they are degraded from the LAMP2A system. LAMP2A is a critical marker for cell-mediated autophagy. In a diseased condition, there is, post-translational modification of this alpha synuclein protein. Why does this happen? We don't know, but because of this post-translational modification like nitration, oxidation, phosphorylation, alpha synuclein is no longer degraded by cell-mediated autophagy. But other substrates of autophagy, they are degraded. So one of the genetic modifications that have been, observed is this A30P and A53P. So A is alanine, P is proline, and T is threonine. So if these mutations happen, if these genetic modifications happen, then we have seen that the degradation of alpha synuclein is blocked. So the same thing is written here. We'll not discuss about it. So alpha synuclein also undergoes macro autophagy. So here, as you can see, these are all the genes like ATG 4, 7, 3, 9, 5. All you have, you have studied about ATG 5 already. So these genes are important for autophagy in the lysosome. So for formation of autophagosomes, these genes are important. So these genes usually they will cause autophagy, but if there is through the mTOR pathway or mTOR independent pathway also. But if alpha synuclein is present, then these pathways, they are dysregulated, and thereby they really do not degrade properly. So this is what happens if there is a alpha synuclein aggregation. So there is not proper degradation of these proteins. So so what happens? Parkin and UCHL1. These are the genes which are important for again. Let me check. Wait. Again, these are ligases and D-ubiquitinases. So these are important for ubiquitination. So if these proteins are also, mutated, then we see familial Parkinson's. So that being said, these are some of the things which happen.
Sir,
So
please repeat the last part.
Parkin and UCHL1, these are ubiquitin ligase and ubiquitin deubiquitinases. I mean, sorry, deubiquitinases. So you have studied about how in ubiquitin proteasome system, how ubiquitin ligase is important, how deubiquitinases are important. If these genes are mutated, it leads to familial Parkinson's. Did you understand that, or is it still confusing?
Not quite, sir. Can you please explain once more?
Okay, for that, I think just let me just, there's a chat. The mode of action in rheumatoid arthritis. So yeah, Shinjon, any question do you have? I think you are not at home yet. So yeah, autophagy is involved there as well. But there are many other functions. We'll talk about this.
So
Wait.
autophagy basically means to like killing the own cells, right?
Yes, yes. That is
So
rheumatoid. There is like immune function involved in that
yeah,
case.
and autophagy is also a immune function, right?
Kind of, yes.
That's why I was bit like
Yes,
I thought like
autophagy is
it was.
also involved there as well. So let's see. But I will have to look into it how exactly what proteins they change. Can you see the slides here?
It is a
Maybe some.
genetic mutation, right? Just last question. For rheumatoid arthritis, it is some kind of genetic mutation along with that, like what will I say, or being autoimmune, it has a mutation in genes.
Yes.
Okay, perfect.
Anything about? Do you remember this slide? Is it visible to everybody?
Yes, sir.
Yes, sir.
So can you remember ubiquitin ligases, ubiquitin and deubiquitinases, which remove the ubiquitins?
Yes, sir.
So yeah, the genes we are talking about here is they involve these I mean ubiquitin ligase and ubiquitin deubiquitinases. So these genes are affected and they cause familial Parkinson's. Let's come back to what we were doing earlier. Is it clear now or is it still difficult to understand? Parkin is a ubiquitin ligase and UCHL1 is a deubiquitinase enzyme.
Yes, sir.
Still confused? So
No, sir. Understood.
what will it cause? It will cause an effect in the ubiquitin proteasome system. That's what exactly is written. So if the ubiquitin proteasome system is affected, then the degradation of alpha synuclein will be affected. So what we are trying to tell you here is that the degradation of alpha-synuclein through multiple pathways, through cell-mediated autophagy, through macroautophagy, and through other ways also. All these things, ubiquitin proteasome system, all these ways, they are not, it is not degraded because of the mutations in the genes. So that's the idea here. Clear on that?
Yes, sir.
So that is what is written here. So macroautophagy is important for lysosomal degradation of oligomeric and aggregated alpha-synuclein, since CMA is unable to handle large protein species. So alpha-synuclein protein levels are significantly elevated in aged human substantia nigra, while both UPS and ALP. So ALP is autophagy lysosome pathway, UPS is ubiquitin proteasome system. All these are for protein degradation. So their activities, they decline with age, so implicating there is a dysfunction in proteolytic system when the disease progresses. So this is the idea. The proteolysis machinery is affected, and that leads to aggregation of alpha-synuclein. So here we can see the ubiquitin proteasome system, how it is affected. Again, the alpha-synuclein, there could be three reasons. Alpha-synuclein is not correctly unfolded, and the proteolytic activity of this 20S subunit, it is blocked. That could be one way. The another thing which could happen is that the alpha-synuclein oligomers, they interact with the lid. This is the lid, the 19S part. It interact with the lid, but it cannot open the lid. That could be one reason. Another thing which could happen is the alpha-synuclein, it blocks the entry into the UPS system. So all this may be a reason why ubiquitin proteasome system does not work properly. Is that clear?
So the so this alpha-synuclein.
Mm-hmm.
Alpha-synuclein accumulates due to the degradation of this UPS system, or rather, the alpha-synuclein is causing degradation of the UPS system. This part is
Again,
I'm
a
not
chicken and egg
understanding.
problem. Not really known, but again, see, since the protein degradation machinery is not efficient, alpha-synuclein means is not. I mean, alpha-synuclein protein is not produced properly. In a normal situation, it will be degraded. If you have a very good, proteasomal, proteostasis machinery. So if you have, let's say, genes for uh, ubiquitin proteasome systems, or if you have genes for cell-mediated autophagy, then the problems of accumulation of alpha-synuclein is lowered. Is that clear? So
Okay.
it is loss
Yes.
of
Yes, sir.
proteostasis on multiple levels. So folding is affected, degradation is affected. All this leads to aggregation of the protein. So where will the protein go?
Okay, that will then aggregate and accumulate.
Yeah. So let's talk about this. This is lysosomal pathway, where lysosome in lysosome, the cell is digested. The proteins are also cleaved off. So there are multiple things which could happen. So the mutant alpha-synuclein, as you can see, it's given in the blue part. This mutant alpha-synuclein, it binds to LAMP2A, and it cannot enter. Basically, the entry is restricted, hence it's not degraded. Then in the B part, you can see that alpha-synuclein aggregates interact with other proteins that are important for survival of the cell. So since they are together with the proteins which are important for survival, they cannot be degraded. A cell cannot, degrade the proteins that are important for survival. Then there could be autophagosomes which could be accumulated. So what is happening? This alpha-synuclein which is mutated, they are getting inside the autophagy machinery, but again the degradation is not happening. So these autophagosomes, they come together, and they remain like that. Another thing which could happen is mitochondrial dysfunction due to aggregation of alpha-synuclein. So there is enhanced mitophagy. So mitophagy is, killing of mitochondria, which is dysfunctional. So if mitophagy happens at an increased rate, that will cause other problems. right? So that could be one of the things which is happening in D. And so these are the different scenarios which could happen. Phagophores, maybe they are not forming. That could also be one of the reasons. So multiple reasons are there for, again, autophagy lysosomal pathway not functioning properly with alpha-synuclein. So we'll only talk about till here. What's the time right now? 8:27, because all of you are probably tired. We'll talk about the later part, later. So one of the most common treatment options for treating Parkinson's disease is giving levodopamine or deep brain stimulation. So if you want to know more about gene therapy trials, there is a link which is given here. We'll be uploading the slides soon. So this is the thing. Also, another problem with Parkinson's disease is there is no biological marker which can tell you whether you have Parkinson's disease or not. So that is a problem. So we cannot really diagnose a Parkinson's disease as such. So again, it is only done based on symptoms. Any questions so far?
Sir, can you repeat the third and please explain the third and fourth slide once more?
Let's go back. Sorry. Wait, let me see. I cannot see the slide numbers. Third and fourth slide. These
Next to
slides, no?
next one, I guess. Next to next one, I guess, means
This
next
one.
to next. The next one, I actually cannot remember the next one. Not this one. Next.
Is this
No,
the
no, no,
one?
no, no. Next.
Next.
Next. I guess. Yes, sir. This
This
one.
one.
This one.
Okay, so you need to read through the entire thing. So what is happening? Alpha synuclein, they are forming, protofibrils. Whatever is given here, the same thing is actually illustrated here. So they are forming protofibrils, and that is again getting accumulated in the Levy bodies. That you can see. Then SNCA gene, it could affect the proteasome, ubiquitin proteasome system, as you can see here. That is also affected. Because of this, ubiquitin proteasome system is affected. That could also be due to Parkin and PINK1 genes, which are involved in ubiquitin ligation and deubiquitination. We'll talk about this in detail. Then these alpha-synuclein proteins, which are ubiquitinated because they are sent for degradation, they can interact with the mitochondria also. So that they can, cause the mitochondria to undergo mitophagy. So what happens is mitochondria is destroyed because of this. So we'll talk about this in detail, actually, how PINK1 Parkin, they lead to mitochondrial degradation. And it is very important in normal cells also. We cannot just knock out these genes because do you know about what is the inheritance of mitochondria? Is it paternal or maternal?
Maternal.
It is maternal.
It is maternal. Maternal. So what happens to the paternal mitochondria?
It gets degraded, I guess.
That happens due to mitophagy. So similarly, there are other examples also. I think, let me think about it. I read about another example like this about mitophagy, which is why it is very important. So we'll talk about it actually in the next class. Erythrocytes, do they have mitochondria? Your red blood cells? Do you have
No,
nucleus?
sir. No, sir. No.
They don't have it. So these red blood cells also during
Lifespan short bole.
their red blood cells also during their maturation, they need to, you know, remove their mitochondria. That happens due to mitophagy. So again, so if there is any problem in the mitochondria, we'll talk about mitochondrial dynamics, how mitochondria, combats with this problem. So we are not going to the slides right away because again we are already overshooting the time. But the thing is, the mitochondria could solve this problem by mitochondrial fission or fusion, where if there is fusion of mitochondria, then there are good genes which are there which will dilute the effect of the problematic genes. And there could be mitochondrial fission also that increases the number of mitochondria. So if there are more number of mitochondria present, then the activity will be higher because the number is higher. So something like that. We'll talk about it. Any questions?
And so the next slide.
Next slide. Yeah, what from here? May I stop the recording, or do you still want to record this?
No, sir, you can stop the recording. Just explain this part once more. Means the degrade